ahead and get it started. So before we get rolling into the formal chat, I am just going to go through a few housekeeping tips, such as the code of conduct, which is nothing you have not heard of before. Simply, we are all here from all across the globe. So let's all just be considerate, compassionate, and have some fun, just keep that vibe going. Also, we have a whole schedule for 24 hours under one sky, and we have about 9 sessions after this one, I believe. So please do check out the schedule when you can on the conference.darksky.org and see, what you want to check in on next. All right. And then also, we have some amazing Under One Sky merch. Our communications team really did it this year, as well as Zoom backgrounds, as you can see mine, which is very cute. But please do check out all of our social media assets and merch, really check it out, support. And that's really it in terms of housekeeping. Let's get this party started. So without further ado, I'm going to start our formal introductions. Welcome to Under One Sky's workshop number 2, as I said, Science for Global Impact. Joining us are Professor Joseph Caruana, astrophysicist at the University of Malta, Dr. Kat Kennedy, sleep and circadian psychologist at the University of Arizona College of Medicine, and a postdoctoral fellow in the Social, Cognitive, and Affective Neuroscience Lab, and Dr. Marko Prokic, Senior Research Associate at the Institute of Biological Research, Sinisa Stankovic, University of Belgrade, Serbia. Together, we'll dive into the latest research on light pollution, highlighting underreported findings, key scientific gaps, and emerging insights that reveal whether further study is needed to better understand and address impacts of artificial light at night. And without further ado, I'm going to pass it on to Professor Joseph.
Hi, everyone. Hello, everyone. So, can you hear me? Yeah, very good. Okay, so let me share my screen. You're going to see some trees first, and here we go. And you should be seeing my slides now. Okay. And I'll do this, and let's get started. Okay, so thank you. Hello, everyone, and thank you for having me. It's
Hey,
a great
Joseph, we don't
pleasure.
see the slides yet.
You
Can
don't
you
see the
share them?
slides? Okay, let me share again. What happened? Share screen. Maybe that. And
There we go. Thank you.
you see them now?
Perfect.
Okay, great. Hi, so thank you for having me. It's a great pleasure. Okay, so something happened now from my end this time around. One sec, what happened? Why do I not see them any longer? 1 second, because I've lost my slides now. Let me. Okay. Okay. I'll stop sharing a sec and we'll do this again. For some reason, I lost my slides. And go back in. Okay. All right. Can you see my slides?
Yep.
Okay, good. Sorry for that. Sorry about that. So thanks for having me. Great pleasure to be among this community. My name is Joseph. I'm an astrophysicist based at the University of Malta. It's very early hours here, so I hope there won't be a lot of yawning. In this presentation, I will focus on a local case study of light pollution, showing how these investigations connect to broader globally relevant questions within the field of light pollution research. In this respect, I should note that a most useful resource, in case you don't know about it, is Dark Sky International's own State of the Science document, which carries an array of open questions in light pollution research. And we'll be making contact also with some of the questions of global relevance posed in that document. So I come from this island nation in the Mediterranean Sea called Malta, which you see circled there. This is a better view. It's actually an archipelago of islands. Malta, Gozo, and Comino. Now, this image here is taken from the International Space Station, and it shows Malta at night, the main island. And what I want to illustrate by this image in particular is that Malta presents an interesting case study because being an island, it is of course surrounded by sea. The nearest landmass is Sicily, around 90 kilometers away to the north. And so by looking at the map, we can appreciate that in this respect, one could say Malta is somewhat isolated. But Malta itself has seen a staggering proliferation of light pollution. For years, there were no checks on this growing problem. And up to the present day, there is no legislation on artificial light at night, with one exception for certain zones, which I'll come to shortly. There aren't even guidelines on light pollution, let alone enforceable rules. There are draft guidelines that have been stuck in draft phase since 2020. Unfortunately, what this means is that when you look, for example, from the sister island of Gozo, where the situation so far is better, towards mainland Malta, you see this hideous glow engulfing Malta at night, as you can see in this image here. This is a textbook example of sky glow. It's like a dome of light over Malta, which represents a huge waste of energy, and the cause is badly designed lighting. There is no design there really, and no control, and that's the principal problem. So when I came back to Malta, I realized this problem had to be looked into seriously and tackled scientifically. There hadn't yet been a concerted effort to quantify the problem. So nearly 10 years ago now, I initiated this research effort to start quantifying the night sky brightness on the archipelago. And I've been fortunate to have many good people working with me on this, including a number of young minds, dedicated and enthusiastic students who did great work. So we employ an instrument, the SQML. Some of you might be familiar with this. It's mounted on a tripod, and we're also using a specially designed hood so that the sensor is protected from glare by ambient light. We take our readings, and then we move on to the next location. And by that, I mean that we lay out a grid over a map of the islands using geographic information system software. In total, we have around 350 cells, and we use the SQML to take measurements within each individual cell to assemble a night sky brightness map for the whole archipelago, which you can see here. Note that there are some significantly darker areas in the smaller islands of Gozo and Comino, those dark blue parts. And that's largely because those areas are more rural and they host what are known as dark sky heritage areas. At least on paper, these sites should be protected. So this is a map from the so-called Gozo and Comino local plan, which includes a number of dark sky heritage areas, which are marked in purple. And when you compare that to our map, you can indeed see that these sites overall are darker than the rest of the islands. So that's an example of a good policy, that had been introduced. These studies allow us to offer recommendations in regard to policy, which we would like to see enacted. They allow you to understand very well quantitatively which areas you should strive to protect, which zones are the worst culprits, and so on. And indeed, we would like to have this part of Malta, for example, protected in a similar way to the dark sky heritage areas of Gozo. So lighting would be banned in that stretch. Of course, that alone is not enough. You need to tackle light pollution as a whole, holistically, across the entire archipelago, because light pollution would still encroach otherwise. And our studies enable us to estimate from what percentage of land the Milky Way is visible. Now, Ours is an empirical study. That is, it's based on actual measurements of the sky, and we estimate that the Milky Way is invisible from around 87% of the island's area, which is quite sad. There's also an agreement with the study of Falki et al. In 2016, in which they estimated that the Milky Way would be invisible from around 89% of Malta. That's a very close agreement. We can also compare our ground-based studies to data collected from space, and that way not only do you cross-check, validate in a way your results, but you can also correlate the increased night sky brightness with specific clusters of light. Bear in mind that these data are related, but they're saying something slightly different. I'll explain briefly what I mean. When you have ground sources emanating light upwards, this is scattered and reflected by the lower atmosphere, and this is what creates sky glow. And an observer on the ground with their ground-based instrument will be measuring that light scattering of the atmosphere and reaching their instrument. But some of the light emanating upwards from these ground sources will also escape into space, and that's the direct measurement that one can make from the International Space Station or a satellite. We can also study changes year by year. For example, this is our map, the one I showed you earlier, and this image now shows the difference registered after a year. So note that this is not a map of the night sky brightness, but a map showing the difference in the night sky brightness between two given years. So a negative value, so green or teal or blue, that means that the sky was found to be darker there, whereas a positive value, yellow, orange, or red, or purple, corresponds to a brightening of the night sky. And this helps us correlate with changes on the ground. So, for example, you see this bright purple spot here that corresponded to the installation of new badly designed LED road lighting, and it was instantly registered by our measurement campaign. So this is a trend that we observe not just in Malta. In many places, light pollution is on the increase. This is an illustrative animation using the MSP satellite data run by NOAA, and satellite measurements likely underestimate the increase in light pollution because they're not very sensitive to blue light. We have also done test studies on even more refined grids, and that really enables us to follow the offending light sources along structures such as arterial roads, for example. And this ties to an important question that we have in light pollution studies. How does the evolution of the night sky brightness evolve regionally, that is on local scales, not on just large scales? You can say you cannot get more local than what I have just shown you, in the sense that here we were literally correlating the night sky brightness with specific roads, for instance. And we need this sort of correlation between source and effect, not only for our own more in-depth understanding, which is hugely important, of course, but also to enable us to go to policymakers and say, hey, look, this practice is really harmful. You should not do this, but rather adopt this approach to mitigate the problem. But to do that, we need to have something tangible to show. And if the policymaker is reasonable and attuned to what science has to say, then they will listen and be more inclined to act. Now, I also wanted to speak to you about a limitation of studies such as our own. This is an image of thin clouds veiling the night sky, and you can see them glowing sort of orange. Now, this was taken from the best dark side that we have, which we're trying to protect. You can see how clouds have an impact, and this is far more pronounced in this next image, taken at the same site. Light from the rest of the islands is reaching this area, reflecting off clouds and illuminating the ground. You may compare the darkness of the sea there with the brightness of the clouds. Now, our night sky brightness measurements are taken during moonless, clear, that is to say, cloudless nights. But that's a best case scenario, of course. There are many days on which the sky is cloudy, if not totally, at least partially. And looking at that photo, you would probably find it hard to believe that this is the same location from where the following images were taken. This is the summer Milky Way, of course. That's a close-up view. That's the fainter part of the Milky Way on the right, so not the core of the Milky Way, with Comet Neowise to the left. And for context, that's a group of people under a starry sky at the same location. It's the same site, simply different sky conditions. Had I not told you they were the same place, you might have thought that they were different locations. This underscores the point I want to make, namely that an important question that has attracted attention is the impact of clouds on the night sky brightness. How much brighter are conditions at night because of clouds? Not just for astronomy, but also in terms of how we and other animals may perceive the nocturnal environment differently when it's cloudy. I also wanted to say something quickly about seasonality. Over 6 years, between 2014 and 2019, the night sky brightness was tracked from a fixed location by one of the co-authors of one of our studies, Mike Nolle. And one can see a seasonality there. In the summer months, the night sky brightness is higher. We kind of expected increased dust particles in the atmosphere over the summer months to be a contributor to this trend, in particular because we get next to no rain in summer, so we don't wash away many of the particles in the atmosphere. Dust particles, aerosols, and so on. But when we looked at particulate data, somewhat surprisingly, we did not find clear evidence of seasonality. So this ties to a question that pertains not just to Malta. What is the correlation between the night sky brightness and air quality, or conversely, air pollution? Okay. Now, when we look at sources of light pollution, we find a wide array of problems. These are mentioned, the ones mentioned here, those are some that we identified in our study, which are common problem sources globally. Some issues are obvious, as are their solutions. So, for example, we all know that applighters like that are very bad. We know that rather than globe lights, we should have fixtures that direct light downwards. And that, moreover, we should control the area over which that light is spread so that we avoid light trespass. We know that excessive lighting is bad, but we also need to go beyond the obvious. As studies continue to emerge, we're finding that entire ecosystems, not just individual species, can suffer from light pollution. You've got species interaction, and you can get a cascade effect as well. By way of example, I point you to a theme issue of the Philosophical Transactions of the Royal Society, published in 2023, that explores this aspect in a collection of studies. And something that was pointed out there is that artificial light does not have to be very bright to disrupt ecology. Even light whose brightness level is lower than that of the full moon can cause problems. So this leads to an important question. Of course, lowering brightness is good, but is it enough, actually? It prompts us to think about parallel approaches, which are probably required, such as curfews and in ecologically sensitive areas, the prohibition of light sources. And I should also mention here that retrofitting road lighting with LEDs has oftentimes spelled a disaster because the expected savings in energy encourages one to employ brighter lighting than required, which may undermine claims of LED lighting being sustainable lighting. And there's another issue that we should address, of course, the color of the lights. In Malta, for example, LED street lights are blue-rich. Now, a common recommendation is to shift to a correlated color temperature, a CCT, that is less than 3000 Kelvin. But something to bear in mind is that by going by CCT alone does not guarantee that you will not still have a short wavelength peak. A CCT below 2700 Kelvin is preferable, and even so, you're not necessarily guaranteeing adequate suppression of the shorter wavelength blue-green radiation. So a question with global import is what measure is best to recommend and use for color criteria, given that limits on CCT are oftentimes insufficient. I will only mention here, as I don't have time to go into the details, two other measures that have emerged. One is the Spectral G Index, which made it into the European Commission's Joint Research Centre guidelines on green public procurement, for instance. To quote from the document, in parks, gardens, or areas considered by the procurer to be ecologically sensitive, or any area within a 30-kilometer radius of an urban optical observatory, or within a 100-kilometer radius of a major optical observatory, the G index shall be equal to or above 2. The other measure is the SP ratio, which stands for scotopic to photopic ratio. In the science of vision, the term photopic refers to our vision in conditions of bright light, whereas scotopic refers to our vision in low light, such as during the night. And the cells in the eye responsible for scotopic vision are most sensitive at shorter wavelengths and in the blue-green region of the spectrum. Lowering this SP ratio is particularly useful to cut down on blue-rich light. To give you an idea, an SP ratio below 1 would be comparable to phosphor-converted amber LEDs. From a light pollution standpoint, the lower the better, of course. And all this leads us to another global question, communicating with industry. How well and clearly is academia communicating and engaging with the lighting industry and professionals working within that industry? In particular, when you look at the message you sometimes get from industry, you realize that its priorities may very well be different. For example, they might care about a lighting fixture that yields better color rendering. In my experience, and I'm sure in many others' experience, light pollution is not on everybody's mind, even when it comes to lighting professionals. We cannot assume that we do not need to be proactive in getting the message across. And that feeds into the question about the perceptions and knowledge of the general public as well. People who attend astronomy events and other nature events are very often very much aware of the issues surrounding light pollution. We are here, after all, because we care. But there are still those who have the mistaken impression that if one mitigates light pollution, then there will be so much darkness in our streets that our security would be compromised and potentially crime would increase, despite this not being borne out by some studies. So rather than a gap in scientific knowledge, in this case, I am talking about a gap between scientific evidence and the public's perception of matters such as safety. That's a global question. How aware is the public about light pollution? And does knowledge correlate with social stratification? That also touches with a similar question in Dark Sky International's own State of the Science report I alluded to earlier. So since we don't have more time, I'll have to stop here. As you've seen, there are many issues that we need to address if we are to keep our nightly window upon our universe open, as I like to say. So keep spreading the message. And as a final note, I am firmly of the belief that we should very much target the policymakers and drive the message home with them at every possible opportunity. In the end, it is the policymakers that have the power to enact regulations to control light pollution. Clear skies, one and all, and thank you very much. And I'll pass on now to Kat. I should this.
Okay, can you all see my slides? Okay, thumbs up. Hi, everybody. I'm so happy to be here. I'm Kat Kennedy. I'm based at the University of Arizona here in Tucson, and I'm a circadian rhythms and sleep physiologist. And so my goal today is really to talk to you about the kind of human side of health and why light pollution at night is not so great for us. So my goal is to kind of tell you about circadian rhythms, touch on some of the latest research that's hot off the press right now, and then hopefully leave you with some things, some sort of actionable steps that you can take away and think about maybe in your own life. So if you've never thought about circadian rhythms in any detail before, let me start by telling you that pretty much every single cell in the human body contains a little clock in the form of genes that make proteins. And these proteins essentially dictate everything that happens in that cell. And they kind of tick along doing their thing and then shut themselves off on approximately a 24-hour basis. And it's kind of remarkable, this sort of mechanism that we have in pretty much every single cell in the body. But they don't tick along at exactly 24 hours. So I'm going to show some QR codes throughout my presentation, and these you can scan, and it's essentially taking you straight to the publications that I'm talking about. So this is showing from a paper that came out about 15 years ago now, the difference in people's internal body clocks. And we've got males in the top panel here and females in the bottom panel here. And along the x-axis, we have time in hours. And so what you can see, if you can see it on my screen, I hope it's not too small, is that in a population of about 150 people, our intrinsic body clock ranged from about 23.5 to 24.5 hours. So it's like an hour difference across the population. There's probably some people who are more extreme than this. And if you can kind of squint, take a step back and look at this, you might be able to notice that the females here on average actually have a shorter circadian rhythm on average than males do. And I'll come back to this idea that females are a little bit more at risk of some of the problems associated with light at night. So because we have this circadian rhythm in our cells throughout the body, kind of ticking along, doing their own things, dictating everything from our metabolism to our immune system, we require some sort of master clock in the brain to keep everything synchronized, right? So you can kind of think of it as like the orchestrator, you know, the sort of conductor of the orchestra. And so this is found in the brain. It's called the suprachiasmatic nucleus in the hypothalamus. You don't really need to remember that. But what you do need to know is that this part of the brain uses light signals from the environment entering the eye and telling these specialized cells in the back of the eye whether it's daytime or nighttime, essentially. In ye olde days, we would have only had the light coming from the sun. And so those blue short light wavelengths, sort of around 450 nanometers, would have been telling the brain, hey, it's daytime. So body, do daytime things. But, and then at nighttime, you know, the absence of light, these cells in the back of the eye are no longer stimulated. And so the brain knows, oh, it's nighttime, so I can do nighttime things. But the problem that we have now is that we're exposed to so much artificial light at night that our brains get a little bit confused. And so the rest of our bodies can get a little bit confused. And we call this term circadian misalignment, which is essentially a discrepancy between what time it is according to the sun and then what's happening inside of our bodies. In a normal situation, we have absence of light at night, and we get the release of melatonin from the pineal gland that sits just below that master clock in the brain. And melatonin is so important for our health. We kind of think of it as this sleepy hormone, and it is, but it also is a really powerful antioxidant. And so kind of like eating your fruits and vegetables, it really helps to sort of combat disease in the body. And so one of the sort of problems that we're having now due to so much light pollution and kind of weakening of this signal to the brain is that we're getting chronic melatonin suppression, and that can be contributing to lots of different diseases. So one of the sort of problems that we see now due to things like, you know, shift work, which is a great example of circadian misalignment, but also really any reason that you might find yourself having a discombobulated sleep-wake schedule or being exposed to too much light at night, you know, caregiving, if you've had a new baby in the household, caring for an elderly parent, if you're having trouble sleeping, whatever it might be, if you're doing that chronically, you may be at a greater risk of some of these kind of scary sounding cardiovascular diseases. And that's what the literature is showing now. There's pretty robust evidence showing that for people who are experiencing circadian misalignment over the long term, they're at a greater risk of disease. It's not going to happen after one night of lost sleep or one night of, you know, flying across the globe or staying up for a conference. But if you're doing this for years on end, it can potentially increase your risk. But there are, of course, lots of different independent sort of what we say risk factors, right, for ultimately what dictates a person's fate. So, you know, how healthy is a person's diet? Are they a smoker? Are they drinking alcohol? And all these other types of things that can play into it. Some hot off the press data. This actually is a paper that just came out last month. I was going to say this month, but we're in November now. Just came out last month. This was a pretty major study that was associating light exposure at night with cardiovascular disease risk. And so what they did in this study was they took personal lighting data from the equivalent of like think of like a research grade Fitbit. So it's a little wrist-worn light sensor. And this is from the UK Biobank cohort, if you're familiar with that. 500,000 individuals in the UK are tracked. And what these researchers did was they collected 13 million hours worth, or they analyzed 13 million hours worth of lighting data from a 10-year period in about 90,000 individuals. And what they did was they broke down the light exposure into percentiles. So essentially, the people exposed to the least amount of light were kind of chunked into this 0 to 50th percentile. And then the people exposed to the brightest light at night were lumped into this 91st to 100th percentile. And what they found was that compared to the people exposed to the darkest nights, those in the brightest light exposure at night, they had a 32% greater risk of coronary artery disease, nearly a 50% greater risk of heart attack, more than 50% greater risk of heart failure, 30% greater risk of atrial fibrillation, and nearly 30% greater risk of stroke, which sounds really scary when you put it like that, but it's also, I think it highlights the importance of thinking about light exposure at night. And as I mentioned before, that chronic melatonin suppression that we're experiencing might be contributing to this. One of the really important things that I wanted to point out is that there are some sex differences that have been noted in the literature and in this study as well. So I think in this study, they found that it was for females specifically, the risk of coronary artery disease and heart failure was greater than males. And this group of researchers have sort of looked at these data from this huge UK biobank cohort. And they've also found that, and this is a point I really want to emphasize, it's not just the sort of bright nights that are the problem for human health and circadian rhythms. It's that we're spending too much time during the daytime in dim light conditions. And so what that's doing is it's sending a weaker signal to our brain. Our brains really need that strong daylight cue in the day and then the kind of absence of light at night to tell us it's nighttime. But what we're doing now is spending so much time in the day in dim office lighting, dim classrooms, etc. And then we're exposed to too much bright light at night. So we have this kind of weakening of the daytime versus nighttime signal to our brain. And this same group of researchers have found that that combination of spending too much time in bright light at night and then dark days, that predicts greater mortality. They found also that when you're spending adequate time outdoors in the day, so essentially, having that bright light exposure, that's associated with better mental health, better overall health. And then just a couple more specific studies. So there's a researcher I work with here at the University of Arizona, Dr. Lauren Hartstein, and she studies early childhood specifically. So she studies kind of circadian rhythms and melatonin in young children. And what she has shown is that when children are kept in dim light conditions and then exposed to bright light an hour before bedtime, their melatonin is delayed by almost an hour. And this is thought to be because children have very clear lenses on their eyes. And so they're actually quite a bit more sensitive than adults are to light at night. So it's something to think about. Parents might not be struggling to sleep with the light, but the children might actually be particularly sensitive. And then there have been a couple studies that have been published over the last few years, essentially just really trying to highlight this sex difference that we see in the literature, which is that women seem to be more sensitive. And it could well be due to the fact that many females have that, and I'm using women and females interchangeably here, but for all intents and purposes, it's females. It's like a physiologic property. It could be because, as I showed on that slide at the beginning, that females tend to have this kind of less than 24-hour circadian rhythm. That might be what's causing this. But it could be some other mechanism that we don't understand yet. But I just wanted to emphasize that as part of where the field is going, is that we're really trying to think about these sex differences. Another article that's hot off the press right now, so this just came out last month as well. This is from the American Heart Association, which it's really exciting to see this come out. The AHA releases these scientific statements every couple of years or so in their different domains of things they care about. So they have, one for stroke, one for obesity, one for hypertension, things like that. And this was a really exciting statement that just got published that essentially is their acknowledgement of circadian rhythms being at the foundation of everything in health, which they really are, because these little clocks that operate in our cells dictate everything that happens in those cells across the day and night. And so this was a really sort of big move for the field, really exciting publication to really just hope that we can get other physiologists in the field studying all kinds of diseases to really be thinking about the timing of when they're initiating their treatments, and the timing of when they're collecting their data, because timing matters significantly. This is one of the really lovely figures from this paper. And what I love about it is it just sort of shows that there are so many different causes for circadian disruption. I'm particularly interested in shift workers, but there are lots of different reasons that we might find ourselves experiencing circadian disruption. And one of these is sort of mistimed eating. So a great tip I like to give people, if you do have to be up really early for like a flight or something, or you're staying up really late for a conference or something like that, if you can restrict your eating to the window that you would normally eat and not eat during the very early morning period or the very late night period, that can be protective. So there are these behaviors that we engage in throughout the day that can affect our circadian rhythms that aren't going through the eyeballs necessarily. So they're not going through that kind of light to the brain mechanism. And here again, this is just showing some of the different processes in the body that our circadian rhythms regulate. So everything from our immune system to how sensitive we are to the carbohydrates in a meal that we consume. So there have been studies showing that, if we have the same identical meal at breakfast, lunch, and dinner, the amount of time that it takes our skeletal muscle cells to absorb the glucose from that meal is much longer when we eat it at dinner time compared to when we eat it at breakfast time. So our circadian rhythms dictate, the process of glucose being taken up into our cells, along with everything else in the body. So the goal is really to think about how we can be using light as an intervention or strategically timed darkness. These are some key takeaways from this paper. So circadian disruption has this, critical role in cardiometabolic health. We've done a pretty good job, I think, as a field of sleep researchers to talk about why getting 7 to 9 hours of sleep is important for health. But, our lab and others have shown that getting regularly timed sleep, so trying to go to bed at the same time every night-ish and wake up at the same time every morning, is really beneficial for the quality of that sleep and your mood the next day. I think we'll, as I alluded to in the previous slide, we'll start seeing lighting and darkness being used as therapeutics, but it's really important to think about how they can be tailored to the individual, because we all have a slightly different body clock, remember? So thinking about how we can have better ways of being able to measure for ourselves what our circadian rhythms are, to know when light exposure or darkness exposure is maybe best suited to us. That would be really helpful. As I mentioned, meal timing, but also movement, these behaviors that we engage in throughout the day, they are really important for regulating our circadian rhythms outside of the brain. As I mentioned, individual, what we call chronotype, which just means your particular body clock, that has to be considered. And I think where the field will be going now is thinking about how we can come up with interventions that drive behavior in a more positive direction. Some of the things that we work on in our lab that I won't go into in much detail. So we are a neuroscience lab and we do a lot of brain imaging. And some of the things that we're trying to work on right now is to really understand what is happening in the brain in these different brain networks when a person is being sleep deprived or experiencing circadian misalignment. If we can find ways of pinpointing, the sort of shift into sleep deprivation and circadian misalignment, that might be a screening tool we can use in future to identify brain markers of when people are becoming fatigued, and that could be a preventative of accidents and things like that. We're also trying to figure out who is most resilient, and there are, as we're seeing, individual differences across the population. And then a sort of fun, fancy sounding hypothesis that a few of us at the university are working on is something called the mind after midnight hypothesis, which is essentially this idea that when we're awake at night, due to our brains becoming a little bit dysregulated, so the prefrontal cortex that's responsible for kind of rational decision making, it becomes a little bit quiet at night. It might leave us vulnerable to be making bad decisions. And so some of us are trying to study that at the university and see how that can affect all different types of behaviors. This is my last slide. I'll leave you with the current expert recommendations from the circadian rhythms field in terms of the lighting that we should be exposed to. So melanopic EDI is essentially light wavelengths that are going to be activating. So in that kind of short blue part of the spectrum, so light wavelengths that'll be activating those cells in the back of the eye. These are the current recommendations, so minimum 250 lux during the day, and then really getting down to less than 1 lux at night, so really wanting it to be as dark as possible while we're sleeping, and ideally a couple hours before bed, to really encourage that melatonin to be released and to be having good consolidated sleep at night. So I'll leave you with that for now, and I will pass it over to Marco.
The time in full mode for my slides and that you can hear me. Good enough. Okay, great. I'm Marko Prokic from the Institute for Biological Research in Stankovic, University of Belgrade, Serbia. So I will be talking a little bit more about the effects of light pollution on amphibians. Amphibians are today largely underrepresented in the research on this topic, even though their urbanization is considered as one of the most important factors that decline the amphibians' populations. There are several life characteristics why amphibians can be sensitive to artificial light at night. More than 90% of all amphibian species are nocturnal, so they do their activities mostly during the evening and in the night. Also, they are frequently living or breeding near or within the human settlements. They have also complex life cycle considering the aquatic larvae and terrestrial adults, meaning that they can be exposed to artificial light at night in two completely different environments. They also have a very low dispersal ability and strong link with the surroundings, meaning that they are not able to efficiently avoid a source of artificial light. And last but not the least, they're currently considered as the most threatened vertebrate group with more than 41% of endangered species. Herein I have summarized the results from the existing literature. We have unfortunately very little number of studies, 43 studies on amphibians, and I must say that the effects of artificial light on amphibians may depend on examined species, life stage, but also on the type of light and intensity that is used in the research. And most of the studies follow some of the behavioral traits. As for the feeding and foraging, effects of artificial light may depend on the phototoxic behavior that species display. In the positive phototoxic species that go towards the light source, they may benefit from the presence of artificial light, as they can easily catch the insects that are gathered around the light source on one hand, but on the other hand, they're easy prey for the other nocturnal predators or can be easily killed or overrun on the roads. Opposite to them, the species that display negative phototoxic behavior and move away from the light source may find difficulties in providing adequate food. They also need to invest more of their energy and time in finding insects. As for the effects on migration, it was observed that during the spring, when the adult individuals migrate, they tend to avoid illuminated areas. This can result in postponed or an ability to find adequate breeding areas. As for the effects on the reproduction, it was shown that the males reduce their mating calls. Also, the complexity and intensity of calls change. They move more frequently, searching for the dark areas that make difficult for females to find them. As for the females, they're less selective when they're choosing the mates. Also, when both sexes are exposed to artificial light in water, they tend to spend lower time and to complete faster their reproduction. This can have long-term effects, as this can negatively affect the quality of offsprings and later the survival of population. As for the other effects, it was reported that individuals that were exposed to artificial light at night may have altered concentrations of corticosterone and testosterone. Those two hormones usually tend to increase in light pollution areas. Also, some individuals suffer from the oxidative stress. There are some effects on expression of melatonin-related genes. Artificial light can slow down regeneration rate in salamanders. Also, can have negative effects on some fitness traits such as body size, survival rate. Also, it can affect the developmental timing, especially during early life stages such as larval stage. So I also identified some research priorities and gaps. I must say that we need more knowledge about the mechanisms through which artificial light at night affects amphibians. For example, we still don't have information on how the artificial light affect melatonin synthesis and concentration. Also about the immune response, there are some suggestions that immune response may be compromised when the individuals are exposed to illuminated areas, but there is no test that show how the individuals respond if they're under some infection or have some parasites. Also, there is we don't have enough information about the reproductive success and also reproductive output. And I must say that our current literature, is very narrow taxonomically and geographically. As you can see, the majority of studies are conducted on neurons, so frogs and toads. Only 4 studies are on tailed amphibians, even though those two groups differ in many life characteristics. For example, tailed amphibians are mostly negative phototoxic. Their larvae are carnivores in comparison to tadpoles that are herbivores. Also, their development and reproductive processes differ significantly. As for the studies, you can see that most of the research were conducted in the Europe or in North America. Indeed, those two regions are under severe light pollution, but they're not rich in amphibian species compared to some other parts, for example, Southeast and South Asia, where you can see that these parts are under severe light pollution, but also are considered as the hotspots of biodiversity for amphibians. Similar, we can say, for some regions of Central and South America, and also we are missing completely data about the Africa and studies on amphibians from this region. One of the gaps is also the absence of spectral and intensity specific responses. We need some comprehensive studies that will compare the effects of different types of light and intensities. In literature, we can often find that most of the studies use very high light intensities that are not environmentally realistic. Also, we are missing some basic information on phototoxic behavior, and the data that we have are mostly from the 70s, the research conducted in the 70s. Other important priorities are the interaction of light pollution with other stressors. Only 4 studies conducted together with the noise pollution. There is a complete missing in the studies that may for example, examine the interaction between light pollution on temperature and chemical pollutants. Those two changes are often related with urbanization. And the last and the most important things is that we are currently missing the mitigation and conservation strategies for amphibians. The current measurements are developed and tested on other species such as insects and bats. So we still don't have feedbacks how those measurements affect the amphibians. I also and my team are working on this topic under the project Call and Light, or the full name Development under Artificial Light at Night, Biological Impact of Light Pollution on Amphibians. The main goal of our project is to examine the biological effects caused by the long-term exposure to two types of LED lights and on two endangered species of crested newts. So the crested newts are belonging to tailed amphibians or salamanders. That is something new in our study. Also, the other novelties are that we are trying to compare the effects of cool and warm LED lights. It was shown that for some birds and mammals, cool LED lights that is more similar to sunlight have more negative effects. So we are trying to test how those two types of lights affect amphibians. We're using the environmentally realistic intensities, 5 lux that corresponds to intensities recorded in most amphibian habitats. Also, 30 lux corresponds to intensity measured under the common street light. We also are following the effects across 2 life stages and 2 environments in aquatic larvae and terrestrial juveniles. And we are trying to determine, besides direct effects, also possible delayed or carryover effects of exposure to light pollution. The parameters that we are following are growth, development, and survival rate. We are determining the effects on morphology, so the body length and body shape of amphibians, on feeding behavior, activity. Also, we are measuring standard metabolic rates and physiological stress. On the top, you can see our model organisms. On the left, it is the newly hatched larva of crested newts. In the middle is the larva that is near the metamorphosis. And on the right is juvenile individuals. We also have some preliminary data from our project. You can see on the graphs that both cool and warm LED lights affect significantly metabolic rates in larvae. They lead to significant increase in metabolic rates. As for the delayed effects, we reported only delayed effects 21 days after exposure in individuals that were exposed to cool LED lights. This type of light also had the greatest effects on activity. They slower developmental rates affected the body size. Also, we reported species-specific response, and the species Triturus ivanbureshi was more affected and more sensitive to light pollution than Triturus macedonicus. And for the end, I must say that our project is funded by the Science Fund of the Republic of Serbia, and it is conducted together with our colleagues from the University of Novi Sad and Leiden University. On the right, you can see our experimental settings. On the tops are shelves with warm LED lights. In the middle are control groups, and at the bottom we have individuals that are exposed to cold LED lights. So thank you for your attention. I also would like to this chance to invite you to our conference and light that will be at the end of November. It is free for registration, and it is a hybrid type of conference, in person or in online. Thank you once again.
That was really, really insightful. I don't know about everyone else, but I sure did learn a lot, from optics to salamanders to circadian rhythm, which honestly is a topic that I cannot get enough of. So now we are going to break out, well, not really break out, but we're going to start our Q&A session with Marco, Joseph, and Kat. So we do have quite a few questions that came in during all of the presentations. Thank you all so much. They were super insightful. So let's get started. All right. So first for Joseph, from Caroline, can you talk more about the tools you use to capture sky quality measurements? I'm especially interested in the hood of your SQM meter.
Right. So we use, as I was saying in the presentation, we use this photometer essentially called SQM, Sky Quality Meter. And there are different varieties of that meter. So one of them, the one that we use mostly, has a lens attached to it. So it comes manufactured with a lens. It's not something that we add to it, but it's actually made with a lens. And that one has a narrower angle. So we use that mostly for our roaming measurements. There's another version of that which has a wide beam, so to speak, and we have made some use of that as well, but for fixed location measurements. And we've also experimented with having a fixed station sort of recording continuous data as well. Now, so that's as far as the sensor is concerned. And then we have, I mentioned this hood that we adopted later on in our research work. So initially we had to be very, very careful about glare. So we had to make sure that the location from where we were taking our measurements wasn't being unduly affected by other light sources, lamps, street lamps or something like that. You had to make sure that it was shielded or far away from such sources. Later on, we adopted also this hood that I mentioned. So we made measurements. Well, first of all, we took into account the opening angle that I was mentioning of the SQM itself. So we take into account the full width at half maximum. So how basically the sensitivity of the sensor as you move out from the center. I mean, they were relatively straightforward calculations, and basing on that opening angle, we also did tests from. So we would move the sensor away from a light source, and we would measure how glare would be affecting that. And we recorded that trend. We did the tests a number of times, also in different conditions, also on the field, not just in a controlled environment, but also out in the field, until we sort of honed in on the best measurements on the best configuration for that hood. So you want to make sure that basically you are not obstructing the field of view of the sensor, but you are removing glare from sources nearby, and we did a number of tests to do that. Yeah.
Wow.
I hope that answered the question.
No, yeah, I think you did, Joseph, because you actually got a lot of questions about your SQM data and SQM specifically and how you use them. And to follow up on exactly like how you used, you know, the SQMs and the hoods, Mike did ask, how do you calibrate the hoods between the SQMs?
No, so actually, to make sure that we didn't have any drift and so on, we were using the same SQM as we were roaming. So that was essentially one person carrying the same SQM with the same hood all over those grid points, just to make sure that, as I said, I mean, in the one I showed, that was actually the first base map that we did. That was when we didn't have the hood yet, so we were very careful about where we were taking the measurements from. And later on, when we started using the hood, and therefore, you know, it simplified life, you could say. The methodology did not change. We were using the same sensor with the same hood, and we would go to different locations using exactly the same setup.
That's actually really good to know. Yeah, I think that really clears it up because I thought it was multiple like different SQM meters as well. So that is actually really wow. All
It was the
right.
same one. A lot of
Right.
work for
I was gonna say that
the
sounds
results.
like a lot of work, but I also didn't want to like, so okay. Wow. Oh my gosh. Well, thanks for clearing that up. All right. The next one goes to Kat. For Kat from Mike, what about adults who've had cataract surgery and new lenses? Does that change how their circadian rhythms are affected by light?
Oh, that's a really good question. I guess it would depend. Yeah, because normally by adulthood, like you kind of have like a lifetime's worth of damage to the lenses. So yeah, in theory, you might be a little bit more sensitive. Yeah, it's a great question. I have to think about that a bit more. But yeah, in theory, yeah, you would be a bit more sensitive.
Yeah, I gotta say, every time I go to the eye doctor, they have something new to tell me about my eyes. So, and it's not usually positive. So
That's why it's important.
literally, yeah. So, oh my gosh. But yeah, that definitely makes sense. And I think that was something mentioned in Lynn People's books in terms of like, as you age, of course, the way light affects you is very different. We become a lot more sensitive. So, yeah. All right, right on. Also for Kat from Todd, can you suggest consumer grade instruments available to measure M-EDI? And I might ask Todd to clarify if you don't know.
Yeah, Melanopic EDI. Yeah, so it's the kind of the light that you're being exposed to. Not so much consumer grade in terms of just if you're trying to measure the light in your home. If you want to get really nerdy about it, you can have a lux meter. You can get lux meters pretty affordably. But there's a lot, I've looked at many lux meters and there's a lot of differences between them in terms of the readout they'll give you. And if you're putting it on the floor compared to where is eye level, it's going to give you a different reading and things. I think you don't need to worry about it too much if you follow a couple rules of thumb, which is just, have the overhead lights on in the day, but turn them off at night and just have like dim table lamps, and that should be fine. You just can't get the brightness to be able to be really activating those systems as long as you've not got, super pure blue light coming in through those light bulbs. So I try and tell people not to worry about it too much, just to have kind of as dim as you can have in the evening that still feels comfortable. But again, what I really want to emphasize is if you're getting enough light during the day, you are protected a little bit against some light at night. It's not to say you want to have like tons of light on at night, but as long as you're getting outside in the day, you're okay with having some, screens on at night and some like lamps on, and that's fine.
That's really good to know. And I'm glad you mentioned that because I live in Washington, as I think I've said like 4 times. But we're going through like the seasonal change with all the gloomy weather. And I just bought this giant, like, oh no, you guys can't see it, but like the happy light,
Yeah, they're great. Yeah.
right? Okay, but now I'm conflicted because it is really bright. It's almost like looking into like a
That's
white
great.
sun.
That's great. And as long as it's not hurting your eyes, like,
Okay.
having that on in the morning, that's great. It's really
Okay.
good. Yeah.
Okay, awesome. So I will keep to turning it on in the morning instead of at night in the evening. Yeah. So awesome. And Chris also says that he loves low light in his house. So shout out to Chris.
There are lots of, you can get really nerdy with different light bulbs that change colors and all the things. And you can go down that route if you're interested in it. But otherwise, I just say simple rule of thumb, overheads during the day, table lamps at night.
All right, perfect. All right. And then we do have a few questions for Marco. Actually, I'm gonna read the one that just came through the chat. Where did it go? Oh my goodness. Marley asked Marco, what are some strategies that are being worked on to mitigate light pollution effects to amphibians?
There are several mitigation efforts that should be prioritized. So we need to reduce sky glow, because it is affecting a lot of amphibians, especially their foraging and mating. Also, one of maybe efficient strategy may be formation of dark zones, because amphibians doesn't have a broad range of movement, so it would be good, nice and enough maybe to form some dark zones around the freshwater habitats such as ponds where they're producing. So we could protect the reproductive process. Also, we could protect the larvae that are developing in ponds, and of course, some using of motion sensors, dim technologies. Also, probably restoring the vegetation that may buffer the urban areas and lighting. And for now, there is suggestion to use of warm spectrum lights, but it is still to test those effects.
That is really great that you mentioned larvae and like mating rituals and things, because I actually learned quite a bit about that in the marine edition of Nightscape. I think it was at the beginning of the year, and it was really just mind blowing how much light impacts marine creatures, you know, and just like habitats like in the water. Um, so that's something to definitely keep like being conscious of because of that reflectivity. I just didn't even think about, you know, brightness levels and like how it like, gosh, pierces the water for lack of a better word. But yeah, for sure.
Completely different environment, terrestrial and freshwater, and also larvae are not available to move from the water. So if there is a light source beneath them, they're forced to be under the light. They have no chances because the ponds are usually small, and the larvae are developing in them, and they're forced to be under the artificial light through their development. So this stage is very sensitive.
And how does that impact the larvae? Do they like not end up hatching at all? Are they born with like defects? How severe is the
One
impact of
study
light on
suggesting
larvae?
that the hatching success is lower. Also, it affects the development. They tend to develop faster, to accelerate their development, so they can move from the stressful conditions. But this brings some costs because their body size is lower, and they have problems in surviving in terrestrial harshest environment when they're metamorphosed. So it have several negative effects.
Yeah, that's a shame.
Yeah.
All right. Well, this is kind of a question for everybody. And we can start off with Joseph and then go to Kat and then go to Marco. But when you're advocating for dark skies, how do you balance sharing science with engaging the public in a way that really resonates with everybody?
I think to some extent you have to adopt different strategies with different groups. So as I was saying before, like if you're organizing an astronomy event, you're going out with telescopes and inviting the public to come and join you. Generally, the people who come to those events are very much interested already, and they probably are already aware. They've done their reading sometimes already. They know that light pollution is bad. You know, so with those people, you can you don't need as much effort, so to speak. They get the message already. They share your passion. They're already on board, so to speak. We do different work. We do a lot of work with different people. So, for example, when we go to schools and we talk to school children, you know, we tailor our message, of course, to the kids. When we speak to policymakers, generally speaking, you take your studies with you and you try to show tangibly what the damage is. So you're saying, okay, as I was saying, I think I alluded to this in my presentation. So you say, okay, look, this is what positive change we can do by just making this tiny bit of change here or there. Look what the positive outcome of that would be. And if you have the data to back that up, all the better, because the policymaker will want to take decision based on data. Now, unfortunately, of course, it's not always the case that the policymaker will listen to what you have to say. There are different pressure groups and so on. But yeah, in general, I would say the message has to be tailored to the audience that you have. There's no one, at least in my experience, I don't think there's one single message that you can use with everyone. You need to read your audience, and you deliver the message according to the audience that you are speaking to.
So I'll just chime in and say, yeah, it really depends, I think, I found on who I'm speaking to. And I have a background in marine biology and environmental science, and then moved into studying humans. And my initial interest was, was bringing the environmental stuff in and trying to get the people to care about it was difficult. And then I was like, okay, well, if I show the people how, having an unhealthy environment affects them, then they understand. So it's about trying to learn the right language with the people that you're talking to, and also storytelling, I think, is the most powerful way of being able to, bring someone around maybe to a new idea.
Yeah, I like that. What about you, Marco?
Still struggling with this because I'm primarily researcher and I don't have a lot of experience in public appearance. So I'm trying to improve myself by trying to explain to different people and people from different backgrounds. So it's not easy for me sometimes to stay on the exact level that people are expecting. So yeah, it is job that I think I need to learn and be a little bit better in this topic, because it is a great problem and we need to talk to everyone and people need to be aware of it and move away from the science and research, try to get to other people so we can try together to contribute to some solutions of light pollution problem, I think. So
I agree with that. And I think you're doing a great job. I mean, you were talking about salamanders and I couldn't relate more, Like I really resonated with that. Just like, a small creature, that can't move away from the light made, my heart cry a little bit. So yeah.
I hope so that the same people are feeling the same. Thank you.
Mm-hmm. All right, a few more questions before our next session. But Ryan asked, and I think this is for everybody, have industry professionals acknowledged your academia's communication efforts regarding light pollution? And we can start with Joseph.
Right. So when it comes to the lighting professionals, sometimes an approach I've adopted is, I mean, because it depends who you get, really. So some do get it. Some are very much already on board with us. However, you do get some who either have their own priorities or perhaps they don't care as much. In those cases, what I've done is, if I know that there's some entity responsible for the installation of light in some location, say, and it's going to, that I know in advance that they're going to do works to install lights at some location, I try to approach that entity rather than the company, say, responsible for the lighting itself, and I make sure that it trickles down from the entity responsible to the supplier of the lighting. So essentially, you lobby the entity. It could be your local council or it could be some agency or something. You go there, you go to the entity responsible for the installation of those lights, and you get the message across to them. And you try and say, okay, look, it's an easy, there are some easy fixes. Let's go for a different spectrum of light. Let's go for slightly less bright luminaires or luminaires that are much less bright in some cases. And then they insist with the supplier, with the industry people to supply that kind of lighting for them. It's not always like that. I mean, there are also, as I said, lighting engineers, for example, that are very much attuned to what we have to say. So yeah, it depends. The answer, it depends. It's not a one size fits all. Sometimes you get people who are receptive. Other times you do get people who are less receptive to the message. So you have to work around that. Yeah.
What about you, Kat? Any thoughts?
On how to sway the lighting professionals? Is that like how to work with them, how to communicate with them?
Yeah, I guess, or other professionals who are outside of the field, of like knowing about light pollution and the impacts.
Yeah, I mean, it's definitely a big push in the circadian field to try and improve lighting in classrooms, to try and improve lighting in offices. Just because, again, going back to this idea that we want the daytime signal when it's daytime, and that helps people at night, people who have fewer sleep problems. There's also, a bunch of people who are trying to work on, and this is more of like a long game, smarter architecture around cities, like urban planning, so that, if you're walking around a city or an urban environment in the daytime, are you surrounded by tall buildings so that you're not getting great kind of solar angle telling us that it's daytime? So there's a lot of circadian people trying to work on making us function better during the day to sleep better at night. And then, yeah, anything we can do, like through what Dark Sky is doing to be preventing all the light at night is helpful. But yeah, just trying to get lots of different people with lots of different skills in the same room is helpful. Experts in different areas, and lots of citizen science, honestly, that's what makes a huge difference. So.
Yeah, totally. Can't agree more there. And Marco, last but not least.
Yeah, we have some plans to work with some industry companies that are developing lighting. So we're trying to test some eco-friendly lights for amphibians. There are some lightings for insects, but I hope we will during the next year start testing their systems and how they are efficient with amphibians, and especially as the ponds are, they're always near the gardens and public spaces. Also in some natural parks you can find lighting, so it will be super cool to see how those systems affect amphibians.
Yeah, I can't wait to see it as well and hear about it too. I'm gonna check in. So yeah, because now I'm hooked. But with that, I do want to say thank you to Joseph, Kat, and Marco for coming and presenting all of your research and experiences in terms of, optics and astrophysics and circadian rhythms and, amphibian research, which, people don't think of amphibians enough. So that's now going to be my new dung beetle. But thank you all so much for joining. I hope you learned a lot. I hope you stay on with us, and I hope to see you at our next session at, I believe, hold on, let me pull up my notes. Okay, I want to get it right. 1 to 2:30 a.m. EST will be our regional story session, Middle East and South Asia, where our advocates from the Middle East and South Asia present local perspectives and initiatives. So it sounds like it's going to be another insightful one. I hope we're all there for it. And let's take a bit of a break. So I will see you guys at the next session. Thank you all so much. Beautiful, wonderful presentations again. Thank you. And I will see y'all soon. Literally, I think 10 minutes.